hand you over to the chairs of the first session, which is largely a genetic session, the genomic perspective of inherited cardiac disease. And I'd like to hand you over to Catherine Mercer and Eleanor Wicks, who are going to take over.
Great. Thank you very much for your introduction, Jan, and welcome everybody to what I'm sure is going to be a really exciting day. There's a lot to get through, so I'd just ask if the speakers could keep to time. And I'll kick off. We're going to start with Dr. Maggie Williams, who's a consultant clinical scientist from the Bristol laboratory. And what she has to tell us about the changes in genomic testing is so important because, as you are all aware, the way in which genomic tests are funded and requested or requested for is really in a huge process of change. So thank you very much. Thank you very much to Maggie.
Good morning, everyone. First of all, I'd like to thank the organisers for giving the cardiac testing labs an update slot at the AICC today, and indeed for giving us the opening slot of the meeting. What an honour. Thank you very much. Many of you will be aware that laboratory genetic services in England have been recently reorganised into 7 genomic laboratory hubs, contracted to deliver a centrally commissioned testing service. This service will operate to common national standards and reporting times, delivering that equity of access we want for our patients. And the 5-year vision is to provide more testing by whole genome sequencing and new opt-in pathways to research, with the aim of building a knowledge base to inform discovery in clinical trials. Cardiology testing will be delivered by 4 specialist cardiac hub laboratories at Oxford, supported by Wessex, Manchester, Brompton and Bristol, each providing for appointed geographies. The hubs are closely aligned with devolved nations' cardiac testing labs to share lessons learned. And this structure provides a working operational network. Meetings so far have been mostly virtual. However, we were able to sample each other's home baking skills at an initial face-to-face gathering. Work themes include, firstly, standardising test protocols, the initial priority being gene panel content. Then standardising variant interpretation and reporting, an important part of which is data sharing, so that reporting decisions are made on the best available information. Then we have coordinated service development to support the new National Test Directory and providing, importantly, appropriate educational support for clinical teams. The Genomic Medicine Service is built around the National Genomic Test Directory, which defines all commissioned genomic tests and the testing that is appropriate for each clinical indication. The directory will be updated annually through a defined process of expert review. And cardiology is a specialist test grouping with available tests listed under indication R codes. There is also some broader testing for acute presentations or syndromic phenotypes, with the involvement of clinical genetic services to support. Cardiology testing is either gene panel based, where a defined set of genes is analysed according to the presenting clinical phenotype and family history, or single gene testing, where the phenotype is more specific. And each test has matching eligibility criteria, which define which patients are eligible for testing and which clinical specialties are able to request, and also the test method. Although it's important to say that centrally commissioned resource is limited, so it's important that testing is targeted to those where it is likely to change patient management or potentially provide the option of cascade testing for relatives in conjunction with clinical genetic services. To share some test directory complexities from our early experience of working with the test directory. For rhizopathy referrals, for those with a mainly cardiac phenotype, the paediatric cardiomyopathy panel is a good option. Where cases have a more syndromic presentation, the R27 whole genome sequence congenital malformations test is more appropriate, and a singleton or trio approach to testing can be adopted depending on the specificity of the phenotype. There is a new indication for idiopathic ventricular fibrillation for survivors of proven cardiac arrest. And for congenital heart disease, we currently have a microarray test and again access to the whole genome sequencing R27 test for syndromic cases. My colleague Julia Baptista will talk later in this session on the utility of the R14 rapid exome test in acutely unwell infants. To make a request, the sample just needs to go to your local GLH, who will arrange all of the onward trafficking to the allocated specialist cardiac hub. But please supply sufficient clinical information to assess eligibility and the R code for the test, and it's also really helpful to know if ICC review has taken place. You can also request a combination of panels for cases that have overlapping phenotypes. And if you're requesting relative testing, please provide as much information on the index case as possible. The cardiac hubs are working together to develop a cardiac specialty request form that will hopefully support you through this requesting process in the future. And the details of contact lines for all of the cardiac hubs are presented on the right of the screen. So really our first success as a genomics community has been to standardise the gene panel content within our laboratories so that all of our patients will receive exactly the same tests regarding of where it's delivered. Gene panel curation involved many of you at this meeting using the PanelApp crowdsourcing tool. Genes are classified using a traffic light system based on the amount of evidence available, with only green genes analysed and reported in clinical practice. All cardiology panel content was agreed in February 20 for a go live in April 20. However, at that point, we had a certain visitation that put a halt on proceedings. Within PanelApp, you can see the gene content for each panel, the evidence for curation, and you can look at the defined panel for the diagnostic service by clicking on the blue Download Signed Off panel button. Despite COVID, the service achieved transition to the 4-hub testing model and PanelApp compliant panels last month. And Genomics England are working with us to review how to make PanelApp more accessible for our clinical users and laboratory users alike. Work is actively ongoing to standardise variant interpretation and reporting. The cardiac lab lead is Dr. Kate Thompson at Oxford, who's working with the ClinGen group to develop cardiac gene-specific variant interpretation guidelines. We're all aware of the benefits of the MYH7-specific guidelines, and this year we are actively awaiting guidelines for core sarcomeric genes relevant to HCM and DCM. The hubs have established a cardiac variant interpretation monthly group with representation from all testing laboratories to compare use of the classification criteria and agree basic principles, particularly where interpretation is challenging. The group has specifically reviewed approaches to managing Titan variants. This case study, kindly shared by Jessica Myrin from the clinical genetics team at Leicester, highlights the benefits of everyone working together. The index case is a 36-year-old DCM male with extensive family history, and the R132 DCM testing showed a Titin truncating variant in the proximal I band. This variant was classified as a VUS based on the limited evidence available. We know there's a general overrepresentation of I-band truncating variants in DCM patients compared with controls. There was an absence of this specific variant in population databases and only one published DCM case. There were 2 ClinVar reports, but unfortunately we were not able to use this information. There was no available phenotypic information available with those reports. Data sharing with the other hub labs revealed another index case in the Northwest hub and 2 further families with this variant with some segregation data. And this evidence enables us to classify this variant as likely pathogenic and will benefit all of the families involved. A national genomics reporting template has been developed by an expert group to facilitate sharing of complex information to patients and specialist clinicians. And the cardiac hub labs are now reporting all testing in this format. There is a clear results summary, implications and recommended actions, with the technical information separated out at the bottom of the report and the evidence for classification clearly presented as an appendix for clarity. 2020 also heralded the very welcome arrival of national guidance on reporting variants of uncertain significance, or as a colleague describes them, very unhelpful statements. And this will hopefully overcome the paradox where labs can potentially over-report to avoid missing anything that might be important in the future, and clinicians then consider that all reported findings must have clinical significance. The guidance advocates only reporting clinically actionable, pathogenic or likely pathogenic findings. Or where we have variants of unsignificance, we only report where additional testing, for example, RNA functional studies or family segregation studies, could reclassify the variant of uncertain significance to likely pathogenic. Occasionally, there'll be certain scenarios where MDT discussion concludes that the VUS is the likely cause for the patient, but unfortunately no follow-up tests are going to be possible, such, for example, in the scenario of adoption. And it would obviously be pragmatic to report these cases also. The real concern of over-reporting variants of uncertain significance is that we are overlooking the real diagnosis for the patient. And it's important that you're all aware of the limitations of genomic testing. There may be as yet undiscovered genes that aren't in our panels. And some regions of genes are difficult to sequence, and as such, there may be gaps in coverage for genes that are listed as being tested. Certain variant types are not detected or could be overlooked with current technologies, and potentially also overlooked with a single testing approach, which is used for most cardiology testing. Detection of mosaicism is also limited by the sensitivity of the specific test employed, and mosaicism may not always be apparent in a blood sample. If you have a really strong clinical hunch that a particular gene is involved, please let the laboratories know. Clinicians are often right. I will leave you with the future work themes for cardiology genomics. Firstly, centralising current and legacy data. We have various options to us at the moment through Decipher or potentially Clinical Variant Arc, which is the new genomics database coming out of the work of the 100,000 Genomes Project and the new Whole Genomes Pathway in the Genomic Medicine Service. We have ongoing refinement of the clinical indications in the gene panels and to find a robust way of updating these on a regular basis and translating those updates into testing in the laboratory. Standardising clinical data is really important to hold that alongside variant data to make the most of genomic data in diagnosis and research. Continued joint working between the AICC, the hubs, clinical teams, NHSE and Genomics England to move the services forward. And building on the legacy of the 100,000 Genomes Project to inform specific application of whole genome sequencing within cardiology. Currently, there are no specific cardiology indications which employ whole genome sequencing, but there's discussion ongoing at the moment for potential candidate indications for WGS in phase 2, and this is something we must all feed into. So I'd like to thank you all for your attention and listening today. Thank my wonderful laboratory colleagues across the Cardiac Laboratory Hub network for their collaboration and enthusiasm in working together, and the clinical teams for their support, particularly to the Bristol Cardiac Laboratory team for their ongoing support in delivering this work at Bristol and for their help with some of these slides. Dr. Ellen Thomas, who's a wonderful clinical advisor working with all of us with a genomics and NHSE hat on. Dr. Emma Baple, who's our clinical rare disease lead in the Southwest, and again, a great support for our services in the hub, and the conference organisers again for giving the laboratories a chance to meet and talk with you today. Thank you very much.
Maggie, thank you very much for that very clear overview of how things have changed, and there is immense change in this area, so that's really instructive at this time. We've just got a moment or two left, and there's a couple of questions. One from Elijah. He says, PanelApp still has some gaps and errors. Is this under review?
Yes, we're in a cycle of review at the moment with PanelApp for comments that were sent in by the end of January, and I believe it's proposed that PanelApp will be reviewed 4 times a year. So please feed in any particular comments either through PanelApp or through your local laboratory hub, and we will do our best to engage and correct and get things in the best possible state for our services.
Great. I think just time for one more perhaps. So Rob Hastings points out that there are specific gene variant guidelines, and you did allude to Kate working on those, incorporating them in the interpretation. So it sounds like that work is going to be ongoing.
Yes, certainly all hubs are using the MYH7 specific guidance, and we're waiting for ratification of the new sarcomeric gene guidance, and then we'll be meeting as a group of hub laboratories to agree how we're going to be using that and roll that out in a standard way for the UK laboratory testing.
Brilliant, thank you for that. I think just one last question. So Pete Constantinou says, great talk, thank you. Are you using a platform such as Whole Exome that will enable iterative reanalysis with improved bioinformatics and identification of novel genes, or custom-built panels to improve capture and coverage?
I think there's a mixture of both going on in the hub labs at the moment. Some are employing defined wet lab panels with the basis that those actually give better coverage and deeper sequencing. Some are using virtual panels off of the Exome platform. And I think this is going to be evolving going forwards, and I think it all depends about the rate of change of panels and new gene discovery, because obviously the more genes that are discovered, the more panels need to be updated, and if you have custom built wet lab panels, that's an awful lot of investment to keep redesigning those panels. So currently the gene content is delivered in a standard way, but the technology for doing that may vary slightly, and I think we're all working together and sharing with each other as to what may be the best approach in the future to make sure we've got the sensitivity to pick up things like CMVs, but we're also responsive and can adapt to the change that's needed to make the service the best it possibly can be.
Super, thank you very much.
Brilliant. Well, I have now great